Hey everybody, this is Eric Kong, and today we'll be taking a look at protein synthesis. Protein synthesis is the creation of proteins, and proteins are essential for everyday processes of the human body. Here we have protein synthesis being broken down into two main steps. One is transcription, and the second is translation. So, in most eukaryotic organisms, protein synthesis will start in the nucleus, and this is where the DNA is stored, right here. Moving on, we have transcription, and DNA usually exists in the form of a double helix, and it will look like this. There will be base pairs on both sides bonded together by hydrogen bonds. And the process actually starts with the enzyme known as Helicase, which I'll draw here. What Helicase basically does is it breaks down the hydrogen bonds between the base pairs and splits the DNA, basically unzipping it. This leaves us with a single strand of DNA here. What happens then is a strand of RNA comes, and this strand is being created by an enzyme known as RNA polymerase. What RNA polymerase does is it starts adding the complementary base pairs of the DNA here. One important distinction to note is that instead of thymine, the base pair, RNA will actually have uracil. Anyways, as this happens, the RNA strand basically leaves the nucleus. As we can see from here, it'll leave the nucleus. The DNA will then be zipped back together and return to its normal stage, and then we move on to the second step, translation. So here you can see the RNA, messenger RNA, and it moves on to translation. And basically what this is the creation of a protein from the information in the DNA. Before we begin, we wanted to look at the simplest unit of a protein, and this is an amino acid. What it'll look like, and this is true for all amino acids, is that I'll have an amino group here, hydrogen atom, a central carbon atom, and this carboxyl group. And then the R group. This is which gives the amino acids their unique properties. Here is a list of the different R groups. You won't be expected to memorize them, but it's important to know the different characteristics that they have based on their R group here. And so as we continue into translation, the RNA strand is actually turned into a polypeptide chain. And how this happens is that the base pairs of the mRNAs, you can see here, go through a ribosome, and the ribosome has a larger subunit here and also a smaller subunit down here. And so basically what happens is this occurs in the pairs of 3 base pairs, by the way. Every 3 base pairs of mRNA that goes through this ribosome will call in a tRNA, and the tRNA will actually hold the amino acid. And so what happens is you can see there's this growing chain of amino acids here, and this is known as a polypeptide. As the mRNA passes through, more tRNA is brought in, and this basically fulfills your primary stage of proteins. And this is what it'll look like, basically. And if we wanted to go a little more in depth, we would look at specifically how the proteins bonded. And if we recall back here, this happens by a process known as dehydration synthesis. And basically, a hydrogen from the amino group from one amino acid will combine with the OH group here, and this will form H2O. H2O leaves this cell, and this binds the carbon and the nitrogen from the 2 amino acids, and it'll look something like this. And this occurs on over and over again, as you can probably imagine. So this brings us to our secondary stage. Our primary stage is basically just the order of amino acids here. Our secondary protein structure has 2 main structures that it can turn into, and this occurs as a result of the hydrogen bonds between the carboxyl group and the hydrogen atom in the amino acids. The first structure is a B-pleated sheet, a beta-plated sheet also known as, and it occurs when the two different strands of polypeptides are in parallel structures. The second is the alpha helix structure, and this occurs when it forms a helical shape similar to a single strand of DNA. As you'll learn later on, biostructure has important factor in determining function, and so while these might be the same hydrogen bonds, the way they're actually structured has a huge impact on their function. So as you can recall, moving on to our tertiary stage, we have the polypeptide chain, and the tertiary stage involves interactions between the different R groups. And so as we learned, the different R groups are what makes the proteins unique. And here we'll cover four main interactions here. In the tertiary stage, we have one, which is van der Waals interaction and hydrophobic interactions. So they're kind of grouped under the same category, and the idea is that the hydrophobic side chains will be repelled simultaneously by water towards one direction here. And what actually happen is, since they're so close to each other, they form a van der Waals interaction, which is just a natural attraction between any form of matter. And so if you imagine aqueous environment, pushing the different R groups together, and then they're being bonded by the van der Waals interaction, which is usually a very weak bond, but it's enough to keep the two R groups together. And as you can see, this will actually pull the amino acids together, and this will kind of define the shape of the protein. Another interaction which is unique to cysteine is a disulfide bond. And cysteine has a sulfur atom on its R group, and what will happen is that the sulfide will actually form a bond with the other sulfide from another cysteine. And this has a similar effect, and it'll pull the two amino acids closer to each other, similarly to this here. Another interaction we can have are covalent bonds, and this occurs when a pair of atoms or molecules exchange electrons, thus forming a bond, and that will have a similar effect, and it'll pull the polypeptide chain towards the covalent bond. Finally, we have hydrogen bonds, and this occurs between slightly negative and positive ends, and what it will do is, similarly to all the other bonds that we've discussed, it will pull the polypeptide chain closer together and the amino acids closer together, thus giving it a defined shape. This brings us to our final stage, which is a quaternary protein structure. Quaternary protein structures basically a combination of multiple different strands of polypeptides form into a single protein. Not all proteins go to the quaternary structure. A lot end at the tertiary stage, but those that do have often very complex shapes and structures, as you can see here. There's four different groups. They have the cyan, the green, purple, yellow, and these all bond together to form a single protein. So this is a basic overview on protein synthesis. There's still a lot more to go in depth, and there's still a lot more you can analyze and interpret from this, but hopefully this helps you